Hi, I'm Andy and this is my poster detailing my research work which investigates the vertebral artery blood supply to the brain and its relationship with cognition across the taxonomic classes Mammalia and Aves. So, it is well known and understood that the brain is a metabolically expensive organ to develop, maintain and evolve, requiring between 20 and 25% of the daily energy intake in humans while only weighing 2% of the total mass. This is very large when compared to the brain's energy requirements in non-human primates, who require between 8 and 10%, and in other mammals, who are between 3 and 5. This expensive brain requires a large blood supply which is achieved through two pairs of arteries, the internal carotid and the vertebral. A posterior-anterior connection through the circle of Willis also allows for this larger volume of blood to circulate throughout, therefore permitting for the maintenance, evolution and expansion of a more complex neuron-dense neocortex. The neocortex being the location in the brain where many higher-order brain functions are thought to originate from. Two landmark studies, one by Baldwin and Bell in 1963 and the other by Ashwini et al. In 2008, demonstrated that mammals typically thought of as less intelligent, such as sheep or ox, had lower rates of mixing between cerebral hemispheres, and also that their brain was primarily supplied by the internal carotid arteries. The vertebral contributed significantly less than they did in humans. So if higher brain complexity arises in part as a product of large total cerebral volume and circulation, then it is possible that vertebral artery blood flow may have a relationship with intelligence. This study aims to assess vertebral artery blood flow to the brain and its relationship with cognition and higher order brain functions across the taxonomic classes Mammalia and Aves. It is predicted that animals that are typically described as more intelligent, like primates and corvids, will display larger transverse foramina and therefore blood flow than those described as less intelligent, such as members of the Bovidae family. The vertebral arteries pass through the transverse foramina in the cervical vertebrae before joining to form the basilar artery and subsequently on to join and become the circle of Willis as mentioned previously. It has been shown that the vertebral arteries are the number one driving force behind transverse foramina shape and by proxy arterial vessel area and blood flow volume. Unfortunately, COVID-19 naturally has resulted in more restricted access to museum specimens that would usually be used in a study of this nature. However, there is a wealth of online 2D and 3D resources which could be a suitable replacement. The pilot study concluded that geometric morphometrics were suitable for this study by comparing traditional measurements with the geometric ones. No significant difference was found between methods, showing that the geometric method was in fact accurate, and therefore I could proceed with analysing online images of cervical vertebrae. To do this, landmark measurements of the right and left transverse foramina, as shown by the blue crosses on this typical C3 to C6 vertebrae in the bottom left-hand corner, will be used along with measurements of the vertebral body, which is illustrated by the green crosses, and the vertebral canal, the yellow crosses, to calculate a size-adjusted average foramina area measurement. This is because while an elephant will naturally have larger transverse foramina and vertebrae than a baboon, in relative terms this may not reveal the whole story. The other part to this methodology is the quantification of higher order brain function and animal cognition. There is a trend in cognition literature to use a single human-centric trait and apply it to a range of species. This is quite often likened to asking a monkey, an elephant and a fish to climb a tree. The monkey will obviously perform better, but that isn't to say it's necessarily better overall, especially when compared with the fish's ability to swim or the elephant's empathetic abilities. Therefore, this project compiles multiple factors commonly used as indicative of higher order brain function and ranks them in a binary system for each specimen for presence or absence. This allows for a much broader approach to cognition, but no research on the individual will result in an absent mark. So far, in this study, 7 cognition factors broadly encompassing aspects of theory of mind, emotional intelligence, and tool manipulation have been reviewed over 18 mammal species and displayed on the linear regression graph on the right-hand side of this poster. Both of these numbers, however, will increase as the project progresses. This preliminary linear regression shows a positive significant difference, p being less than 0.01, between this total assigned rank and the adjusted transverse foramina area, therefore suggesting that the larger the average foramina area, and as discussed earlier by proxy blood flow and vessel diameter, the more higher order brain function factors that specimen displays. However, the current R-squared value of 0.3514 shows that this model does not explain all the variation observed within the dataset. This is something to monitor and look at as the project progresses, but could likely be due to other factors such as typical species size or intraspecies variation. Going forward, the primary aim of this project is to include the taxonomic class Aves, with specific comparisons with the typically highly cognitively advanced corvids. As well as this, more mammalian species and more cognition factors such as metacognition or complex sociality will also be added into the analysis. The adjusted transverse foramina area can also be compared against other cognition measures commonly used within the literature. This includes the encephalization quotient, relative brain size, and cortical neuron count, and shall all be sourced from primary literature sources. This will hopefully give more results that enforce what has already been found and provide more evidence towards the prediction that vertebral artery blood supply significantly affects animal cognition. Thank you for listening, and if you have any questions, please do not hesitate to get in touch. I would also just like to say a huge thank you to everyone who is supporting this project and providing me with the specimens required to carry it out. Thank you.